I am Eirini Papapetrou, and I am a professor of oncological sciences and a professor of medicine at the Icahn School of Medicine at Mount Sinai. I am also the director of the Center for Advancement of Blood Cancer Therapies here at Sinai.
Thank you. How did the research grant funding you received from AAMDSIF impact your career?
The research grant from the AAMDS Foundation came at an early time in my career when I was an assistant professor and had only started my independent lab just within the past year. So it really provided us with this initial boost of confidence and funding, of course, that we needed to generate some of our first data that would then allow us to apply for bigger grants. And indeed, this was instrumental as we were able to grow our research program, gather more evidence that we are onto something, and preliminary results. And then we were very successful in securing additional funding, including big grants from the NIH.
What do you consider to be your most notable contribution to MDS research, and why?
So what my lab is known for is the establishment of human-induced pluripotent stem cells, or iPSCs, as models of MDS and AML. When I started my lab, iPSCs were a very new technology, and I and others had used it already to model and study inherited genetic disorders caused by germline mutations, such as thalassemia and sickle cell disease, and also including inherited bone marrow failure syndromes like Fanconi anemia, Shwachman-Diamond syndrome, and others. But while most users of iPSCs were focused on monogenic disorders, when I was getting ready to start my independent research, I had the idea that why not use iPSCs to study blood diseases caused by acquired somatic mutations like MDS and other sporadic blood cancers? Since cancers are primarily genetic diseases, and so I thought that creating faithful genetic models of these diseases with iPSCs would give us a valuable new tool. And so iPSCs are really fantastic cells, and iPSC models give us some unique advantages, as they are at a sweet spot between primary ex vivo patient cells and cell lines, in some ways combining the best of both worlds. On one hand, they come directly from patient cells, but then on the other, they can be maintained indefinitely like cell lines at a pluripotent state. But then we can push them to exit the stage of pluripotency and differentiate to make the cells that we want to study, in our case, hematopoietic cells, blood cells. And when we do this, these cells now resemble in many ways the starting cells of the patient. But unlike the cells that come straight from the patient, with iPSCs, we can do much more. We can do extensive experiments, we can genetically manipulate them, we can scale up our experiments, and we have unlimited cells to repeat experiments as many times as we want to really feel confident in our results. So now, almost 10 years later, we have created many iPSC models of MDS and AML, and we have shown how useful they can be. And we have shared these iPSC lines and tools that we developed with the scientific community and keep sharing them continuously. And we have started many collaborations to enable other investigators to use these tools that we created for their own research. We have also recently established here a new center here at the Icahn School of Medicine at Mount Sinai, the Center for Advancement of Blood Cancer Therapies, whose mission is to accelerate research into blood diseases and blood cancers. And as part of this, we are creating shared resources and facilities to spread the reach of iPSC technology to other researchers within and outside our institution in both academia and in industry.
And what innovative research is being conducted in your lab today?
So we have by now created many models of genetic drivers and combinations, and we are using them to study how these drive MDS and AML and how they determine drug responses or drug resistance. We have identified some promising new therapeutic targets, specifically for MDS with splicing factor mutations, that we are now validating and further exploring for the therapeutic potential. We are also very interested in clonal hematopoiesis, a mostly benign condition in the blood of healthy individuals that increases with age, but that can, however, predispose a small percentage of these individuals to develop MDS.
And how do you support young investigators, both in your lab and in the broader field of bone marrow failure disease research?
One major initiative that we have started in the past few years is an annual workshop on iPSC-derived hematopoiesis, where we gather world leaders from the areas of iPSC disease modeling, cell therapies, but also development, and their trainees. And we provide many opportunities to young investigators to learn about the most exciting research in this area, to present their own work, even if it is not advanced enough to be presented in bigger and more general interest conferences, and also to meet and network with other senior and junior investigators and other trainees.
And this year, as our AAMDSIF is celebrating our 40th anniversary and looking forward to continuing our mission of providing answer, support, and hope, what is your wish for the MDS research space and for MDS patients?
MDS patients have been and still are, for the most part, underserved. We do not yet have good solutions for them. And we need to correct this injustice and do better and develop better and less toxic treatments for them that will not only help alleviate the symptoms of the disease, but provide a cure, or even better, prevent the disease from progressing to a severe disease stage. We and many other investigators are truly committed to conduct groundbreaking research to advance science towards these goals.